Welcome back to my channel. So we discussed about the carbohydrates in the previous part, where we discussed that carbohydrates are basically the polyhydroxyaldehydes or ketones, and we discussed that it has been classified into different categories, the first being monosaccharides, then we discussed that disaccharides, and then we have oligosaccharides, and then we have polysaccharides. So let's just look at the category monosaccharides in detail now. Now, monosaccharide basically means the carbohydrates that are the simplest types of carbohydrates. That is why they are also known as simple sugars. Now, if we look at the structure of monosaccharides, they are the simple sugars, and it is the simple monomeric unit, one monomeric unit, which is going to join together in different ways to form the next classes of the carbohydrate, that is disaccharide, oligosaccharides, and polysaccharides. Now, these monosaccharides have been classified further into two categories. So the first category is aldose, as the name suggests. So aldose is basically a monosaccharide that consists of an aldehyde group in the structure. On the other hand, when we take the example of ketose, the other class of monosaccharide, the ketose, as the name suggests, it is a monosaccharide that will contain a ketone group in its structure. Now let's look at the important monosaccharides that we discussed. So first is glucose. If you look at the structure of glucose, you will find that there are 6 carbon atoms present in the glucose molecule. Carbon number 1, then 2, 3, 4, 5, and 6. And on these carbons, you can find many hydroxy groups present. You can see that this is OH group, this is again OH group, OH group, OH group, and OH group. So all these OH groups are basically justifying what we learned in the definition of the carbohydrates, polyhydroxy, right? So there are a lot of hydroxy groups present on the structure of the glucose. Now, other than that, you can find that at the carbon number 1, there is an aldehyde group present. What does this aldehyde group tells us? This tells us that glucose is an aldose. Okay. So glucose is an aldose that we have to memorize. So if someone asks you that define the structure of glucose, you will say that glucose is a hexose. Hexose basically means a carbohydrate, a monosaccharide that contains 6 carbon atoms. Then you will say that it is a 6 carbon atom molecule which have a lot of OH groups present on its structure, and other than that, it has an aldehyde group present on its structure, which means that glucose is an aldose. Now coming on to the next structure, that is galactose. If you look carefully at the structure of galactose and compare it with the structure of glucose, you will find that it is very similar. There is a lot of similarity between glucose and galactose, and they are differing from each other only around one carbon atom, and that carbon atom is the carbon number 4. So everything is same when we compare glucose and galactose, but at the carbon number 4, the orientation of the hydroxy group and the hydrogen atom, they have been interchanged. In the glucose structure, you see that the hydroxy group is present on the right side of the carbon number 4, whereas in case of galactose, the OH group is present on the left side. So that is the only difference. And most importantly, the structures that differ from each other only around one carbon atom are known as epimers. So if someone asks you that, okay, define epimer, you will say that epimers are the structures that differ from each other only around one carbon atom. And if someone asks you, okay, give me example of epimers, you will say that glucose and galactose are the epimers of each other, which differ from each other only around one carbon atom, that is the carbon number 4. Okay, now coming on to the next example, that is fructose. If you look at the structure of the fructose, you will again find that this is again a hexose. Hexose means 6 carbon atom containing monosaccharide. 1, 2, 3, 4, 5, 6. But the difference is that in case of fructose, at the carbon number 2, there is a ketone group present. Now there is no aldehyde group present. So we won't call it as aldose. We will rather call it ketose. So fructose is a ketose. So glucose and galactose are the aldose monosaccharides, whereas the fructose is the ketose monosaccharide. So this we have to memorize. Okay. Now let's just look at the structure of each monosaccharide in detail, along with the straight chain structure and the cyclic structure. So first, let's talk about glucose. Now, glucose is the most important monosaccharide. I always ask my students to memorize the structure of glucose very carefully, because this is the one structure that is going to help you in understanding the metabolic pathways very nicely, especially the glycolysis pathway. So glucose is the most abundant hexose in our diet. So we take a lot of carbohydrates through our diet, and out of all those carbohydrates that we consume, glucose is the most important one, and whatever the carbohydrate we consume ultimately gets converted into glucose, and that glucose is then metabolized by the body, by the cells, for obtaining energy by the process of glycolysis. Now, glucose is also the building block for complex carbohydrates. Now, what do you mean by complex carbohydrates? Basically, the polysaccharides that we have to look after a few slides. Now, they are also the components of the disaccharides, right? Obviously, this is a monosaccharide, and this is going to be the constituent for the disaccharides. And then they are also found in the polysaccharides, obviously, again, because polysaccharides are the long chains of the monosaccharide molecule. So glucose is one such important monosaccharide that contributes in the building of the long chains of the polysaccharides, and most importantly, starch, cellulose, and glycogen. Now let's look at the structure. Every monosaccharide is having two kinds of structure. One is the straight chain structure, which is basically known as the Fischer projections, and one is the cyclic structure, that is the Hebert projections. So when this Fischer projection, the straight chain structure that we just discussed in the previous slide, this gets cyclized, it gets converted into this kind of structure. So this is the cyclic structure. This gets cyclized, and then cyclic structure known as the Hebert projection is formed. So either of the two are correct, but mostly when we understand metabolic pathways, we often look at the cyclic structures because it's easier to understand. Next we have fructose. Now fructose is the sweetest of all the carbohydrates. You know, we eat a lot of fruits, right? Why do they're sweet? They are sweet because of the presence of fructose in that. That is the reason. Now, they are also a component of disaccharide sucrose. So when we look at the structures of the disaccharides, we will see that fructose is present in the disaccharide sucrose. And as we just discussed, that it is a keto sugar. Why? Because it contains a ketone group. Now, when this fructose, which is a single molecule, gets converted into its cyclic form, it looks like this. So this you have to see. Now, most of the students, they often confuse that sucrose is a pentose. They often write that yes, fructose contains 5 carbons. Why? Because if you look at the structure of the fructose on a whole, if you just have a rough look on it, you will see that it contains 5 carbons. 1, 2, 3, 4, 5, something like this. Students often mistake that. But no, this fructose is not a 5 carbon molecule, but it is rather a 6 carbon molecule. How? See, 1, 2, 3, 4, 5, and 6. So there are a total of 6 carbons. So this you have to memorize. Now, next, let's just see the next structure of monosaccharides, that is galactose. So galactose is found in the disaccharide lactose, that we will see when we will learn about the disaccharides in the coming slide. Now, it is found in the cellular membranes of the brain and the nervous system. And galactose is the C4 epimer of glucose that we discussed. So when this galactose gets cyclized and the cyclic structure is formed, this is how the galactose looks. So both of the structures are correct. One is the cyclic structure, one is the straight chain structure. Now let's just move on to understand disaccharides. Now disaccharides, I just told you that we are going to discuss about 3 disaccharides, important one, maltose, lactose, and the third one was sucrose. So let's just see the maltose first. So maltose is also known as malt sugar, and it is used in the cereals, candies, and brewing of the beverages. Now, if we look at the structure of the maltose, we will find that this maltose is composed of 2 glucose molecules. So this is also a glucose molecule, and this is also a glucose molecule. And when these 2 glucose molecules are bonded together by a bond, it is known as maltose. Now, before we can understand this bonding, we have to understand that the monosaccharides, they join together by a certain bond. And what is this bond called? This bond is known as glycosidic bond. Okay, so the bond by which 2 monosaccharide units, 2 adjacent monosaccharide units are bound to each other is known as glycosidic bond, or it is also known as glycosidic linkage. So this is the name of the bond that is present between the monosaccharide units. So this is a glycosidic linkage which is present between the 2 monosaccharide units. So looking at the structure of the maltose, this is a glucose molecule and this is also a glucose molecule, and the linkage that is present between these two glucose molecules in maltose is known as alpha 1,4 glycosidic linkage. Now why alpha 1,4? Let's understand that what is alpha. So alpha linkage is basically the linkage that is present between the groups present on one side of the plane. So you can see that the groups present on one side of the plane are making a bond with each other. That is why it is being called as alpha. And why it is being called 1,4? Because the carbon number 1 of the first glucose molecule is making a bond with the carbon number 4 of the next glucose molecule. So that is why it is called alpha 1,4 glycosidic linkage. So till now we understood that maltose is a disaccharide, which means that it contains two monosaccharide units. And what are these two monosaccharide units? Glucose and glucose. So two glucose molecules are binding to each other. And what is the linkage present between the monosaccharide units of the maltose? The linkage is alpha 1,4 glycosidic linkage. So let's move on to understand lactose now. Now lactose is also known as milk sugar. Now that sweet taste, the inherent sweetness that we have in the milk when we consume it without adding any extra sugar, you can see that it is having an inherent taste of sugar, right? Sweetness is there. Why is it? It is because of the presence of lactose. So lactose is found in the milk and the milk products. And this lactose is formed of two monosaccharides, galactose and glucose. So this is galactose, this is glucose, and when these two bind together by a glycosidic linkage, it is known as lactose. And the linkage that is present between the monosaccharide units of the lactose is beta 1,4 linkage. Now why beta? It is because the groups present on the opposite side of the plane are making a bond with each other. That is why it is being called a beta. And the carbon number 1 of the first monosaccharide unit, that is galactose, the carbon number 1 is making a bond with the carbon number 4 of the next molecule, that is glucose. So that is why it is being called as beta 1,4 glycosidic linkage. So till now in lactose we learned that lactose is a disaccharide present in the milk and milk products, and it is composed of galactose and glucose, and the bond that is present between these two molecules in the lactose is beta 1,4 glycosidic linkage. Now coming on to the sucrose. Sucrose is also known as table sugar. Now what is table sugar? Table sugar is the normal sugar that we have in a household, which we add to different kind of things. So this is basically a product of sugar cane and sugar beets. This sucrose is made up of glucose and fructose. And the linkage that is present between them is the alpha 1,2 glycosidic linkage, which means that carbon number 1 of the glucose is getting bonded to the carbon number 2 of the fructose. Okay. So in sucrose, we learned that sucrose is a disaccharide and it is obtained from sugarcane, and it is composed of glucose and fructose, in which the glucose and fructose are bonded with each other by alpha 1,2 glycosidic linkage. Okay, so this was about disaccharides. Now let's move on to polysaccharides. Now, as I just told you, that polysaccharides are the carbohydrates that are composed of more than 10 monosaccharide units. So polysaccharides will be having more than 10 monosaccharide units present in its structure. Okay. As I just told you when I was giving the example of bracelet, right? So there is one monomeric unit, which is the monosaccharide, and when these monosaccharides, they join together to form long chains, particularly more than 10 monosaccharide units, when they join together to form long chain, that is known as polysaccharide. Now, these polysaccharides are called glycans. They are not called sugars. So this is an exception. Polysaccharides are not categorized under sugars because they are not sweet in taste, but they are rather known as glycans. Now, these polysaccharides have been classified further into two categories. The first category is homopolysaccharides and the second category is heteropolysaccharides. Now, what do you mean by homo? Homo basically means same. Okay, so homopolysaccharides are the polysaccharides that will contain the monosaccharides of same type. For example, let's say glucose. So let's say this is glucose and this glucose is repeating. So glucose, 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 glucose. So all these glucose, these molecules are repeating unit and they are making long chain. So that will be known as a homopolysaccharide. Whereas in case of heteropolysaccharides, hetero means different. So heteropolysaccharides will have different kinds of monomeric units present in the structure. For example, let's say glucose and then lactose. Galactose. Glucose, galactose, glucose, galactose, glucose, galactose, glucose, galactose, something like this. So there is not only one single type of monosaccharide present, but rather different types. Okay. Let's take the example of bracelet again to understand this better. Let's say this red color symbolizes glucose. So glucose, 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 and so on. So this is a homopolysaccharide. Whereas in case of heteropolysaccharides, what will happen? This is a glucose, let's say, and this yellow is, let's say, fructose. I'm just giving you a hypothetical example just to make you understand. And let's say this blue colored is your galactose, and these things are going to repeat: glucose, then again fructose, then again galactose, and so on. Again there will be glucose, then again there will be fructose, and then again there will be galactose, and so on. Okay. So for example, if you compare it with the bracelets, right? So you have, let's say, a bracelet which contains all the red colored beads. So you can imagine it like a homopolysaccharide. So these all are red colored beads in the bracelet. But if you imagine a bracelet in which there are different kinds of beads, for example, red, then is yellow, then is blue, then again red, yellow, blue, like that. So that is a heteropolysaccharide. Now the examples of homopolysaccharides are starch, glycogen, cellulose, and pectin. So all these examples that are listed here on the screen right now are the homopolysaccharides, and that they all are made up of glucose only. That's a coincidence that these all homopolysaccharides that are listed on the screen right now, they all are made up of glucose. Now you might ask that, okay, if they are all made up of glucose, then how are they different from each other? They are different from each other on the basis of the bondings and the branchings. Okay, so that we will see. On the other hand, if we look at the structure of the heteropolysaccharides, the examples are hyaluronic acid, chondroitin, and then we have vegetable gums. So let's look at the structure of the polysaccharides now, specifically the homopolysaccharides. So we have three examples that we have to discuss in detail: starch, glycogen, and cellulose. So if we look at the structure of this starch is basically composed of two different molecules. The first one is the amylose and the second one is the amylopectin, which means that amylose and amylopectin combine together to form starch. Now, amylose is a linear structure, whereas amylopectin is a branched structure. Now, all these boxes that you can see, these boxes, they are nothing but glucose molecules. Okay, so this you have to take care. So amylose is a linear structure. The glucose molecules, they're bonded together to form a linear structure. So this is amylose. Whereas in case of amylopectin, you can observe that there are certain branchings occurring. So these are the branchings that you can observe. So this is amylopectin. Now this amylose and amylopectin, they make an amalgamation and they collectively are known as starch. So there is a particular composition of amylose and there is a particular composition of amylopectin present in the starch. Now coming on to the structure of the glycogen, you can observe that the structure of glycogen is looking very, very similar to that of the amylopectin. But the only difference you can observe is that this glycogen is highly branched. So this is having more branching as compared to the amylopectin. It is a more complex structure than amylopectin. Now, if we look at the structure of cellulose, cellulose is also a linear structure, and it is having multiple linear structures of the glucose molecules, and all these linear chains are bonded with each other via some bonds. So that is what the cellulose is. So it basically acts like a mesh. So mesh kind of structure is formed where a lot of linear chains are joined together. So we will still refer to it as linear. Now, cellulose is the major structural material of the plant cells, and it is present in the cell wall. So you know that this is the outermost membrane of the plant cell, that is the cell wall, and the inside of the cell wall is the plasma membrane or the cell membrane. So this is the plasma membrane and this is the cell wall. So the cell wall is composed of cellulose. And if we look at the detailed structure of cellulose, you can see that it consists of a lot of glucose molecules. This is glucose, this is glucose, this is also glucose, this is also glucose, glucose, and so on. Here, after this particular point also there are a lot of glucose, and before this point also there are a lot of glucose molecules. Now, the difference is that the glucose molecules that are present inside the cellulose, they are joined in a fashion. So for example, let's say this glucose is in the upside and the next glucose is going to be flipped. If you observe it carefully, you can see that the adjacent glucose molecules in the cellulose are flipped. So if one glucose molecule is straight, then the next glucose molecule is going to be flipped, something like this. And then the next one is going to be straight, the other one is flipped, and so on. And the linkage that is present between the two adjacent glucose molecules is the beta-1,4 glycosidic bond. So this you have to memorize, beta-1,4 glycosidic linkage. Now coming on to the structure of starch, starch is the storage form of glucose in case of plants. So if someone asks you that what is starch, you will say that it is a storage form of energy, or you can say glucose in plants. So in plants, the energy is stored, or the glucose is stored in the form of starch for obtaining energy for the future use. So as I just discussed in the previous slides, that starch is basically composed of amylose and amylopectin. The amylose is basically a linear structure. Yes, this is linear, and the amylopectin is a branched structure. Now, the important thing that we have to consider is that the linkages, in case of amylose, which is a linear structure, the bonds that are present between the adjacent glucose molecules is alpha-1,4 glycosidic linkage. So this is important. Whereas in case of amylopectin, all the glucose molecules, the ones that I'm circling, these are all alpha-1,4 linkage. But at the point where branching is occurring, wherever the branching is occurring, at that point, alpha-1,6 linkage will be there. So let's go back to the slide that I mentioned. So if you see here, you can see that this is the one that I'm marking. It is linear, right? So here all the bondings are going to be alpha-1,4 linkage. But at the branch points, the ones that I'm circling with black. So this is a branch point, this is a branch point, this is also a branch point, branch, branch. So at all these branching points, alpha-1,6 linkage will be there. And again, after all the subsequent glucose molecules, after the branching also, there is going to be alpha-1,4 linkage. So you can see that at the branch point, there is alpha-1,6 linkage, whereas in between the linear chain, alpha-1,4 linkage is there. So if someone asks you that what are the different kinds of linkages that are present inside starch, you will say that in case of amylose, which is a linear chain, it consists of only alpha-1,4 glycosidic linkages. Whereas in case of amylopectin, in the linear structure, the glucose molecules are bonded with alpha-1,4 glycosidic linkages, whereas at the branch point, alpha-1,6 linkage exists. Now coming on to the last example of the polysaccharides, glycogen. So glycogen is also known as an animal starch, and it is the storage form of glucose in case of animals. So all the animals, they store the glycogen in the form of glycogen, they store glucose in the form of glycogen and in the liver. So all the livers that we have, all the different animals, they have liver. In that liver, the glycogen is stored, which is basically the storage form of glucose. And it is also stored in muscles. Now, I told you that starch and glycogen, they look very similar, but the only difference is that glycogen is more branched. It is highly branched. So the kind of linkages that are present in the starch, the same kind of linkages will be present inside the glycogen, but the only difference is that there will be more branching. So at the branch points, we will see alpha-1,6 glycosidic linkage, whereas in the linear chain itself, all the linkages are going to be alpha-1,4. So that's about the carbohydrates. So we discussed about the definition of the carbohydrates, that is, they are polyhydroxyaldehydes and ketones, and we looked at some of the important features of them. Then we understood that it has been classified into different categories, and we looked onto each class in detail and tried to understand the structure in detail. So thank you so much. In the coming lectures, we will also discuss about the functions of carbohydrates.